There'll be 25 multiple guests. I might tweak it one more time. There'll be 25 matching in 3 different sections, right? One section is standard growth curve, one is all that DNA and RNA stuff, and one is just the mechanism of action for chemicals. Right. Those, to me, those are kind of the harder concepts, so I decided to make them matching. The others are just going to be multiple, and you know how I like to write multiple choices. I want you to think about things, right? So when 5% sheep red blood cells are added to a medium, allowing clinicians to discern whether bacteria can destroy blood, the medium is considered what? A selective and enriched, a non-living, a differential, A and B, C and B, or D and B? Which one? It's going to be B, enriched, because you added blood, and it's going to be D, differential, right? So it's going to be B and D, which is going to be AC. You'd have to mark in AC. This is an actual live question. It's probably be on the exam. You'd be surprised how many things I'm showing you that might be on the exam. Do not tell my boss, he's in the house. Harlow, don't go over there and say Pro V is easy. He's getting us all the answers. Sterilization of bacteria may be accomplished by the following processes, except one of these things doesn't sterilize bacteria. Which one doesn't?
Oh, is it pasteurization?
Which one?
A?
A is pasteurization, that's correct. That is
That was
absolutely
me.
I know, because it was some of you, but it's pasteurization is a sanitation method, it's not
It
sterilization.
does not kill endospores.
That is correct. Good. Here we go. A microorganism that grows optimally in our body. This is from the lecture that I sent you. We're supposed to look at. And what's the answer? A mesophile.
Is this the one you sent last night?
Oh no, I sent this one
Oh,
3
okay.
weeks ago.
Just making sure.
Okay.
Because I saw one last night.
Well, I sent 3 things last night. I sent the video on DNA replication. I sent the video on protein synthesis. And I sent you the little chart that I like to use, a little graph that has all the answers on it. That's on the exam.
Yeah.
It is on the exam. I'm telling you, it's on the exam.
I got it on video.
Okay, good. You should get it. Detergents with a 10-minute contact time kill microorganisms by.
Ooh, that's a. Is it A?
Not
D?
D is correct. It solubilizes plasma membrane. Remember, we said that the wash cycle was 10 minutes because that's how long it takes for the detergents to break apart the plasma membrane. Right. Here we go. Which of the following describes flash pasteurization?
71. Is it like?
Which A, B, C, D, or E?
71, 72 for 15 seconds.
Correctamundo. Man, you're gonna do well on this exam.
But you put 71 during lecture.
I know, but it's 71.8 or something like that. So I made it 72.
Okay.
I just rounded it. Okay? All right, here we go. AUG codon functions in coding for amino acid methionine and as a.
Stop.
No, start, not stop. Start codon. The stops are what? UAG, UGA, and UAA. All right, here we go. I didn't put any fill in the blank, but I have some fill in the blank as extra credit currently. Would you like me to leave them in as extra credit?
Yeah.
So, if you know the exact ingredients of meat, it's called what? Chemically defined. In general, something is a sanitizer that is toxic and should never be used on the skin or mucous membrane.
Hydrogen peroxide.
Yeah, no. I mean, yes, but I'm not asking for an individual. I'm asking a group. But I like the excitement. A disinfectant. A disinfectant. Okay, here we go. Something is a term used to describe 2 microorganisms growing in an environment where one of the microorganisms benefits from the relationship, but the other is neither harmed nor benefited from the relationship. This is in the video that I sent that you were supposed to look at. No, not, well, yes, but the specific type of symbiosis. There are three of them. Shall I give a multiple choice? Here we go. Mutualistic, parasitic, or commensalistic? Commensalistic. Commensalistic means one of them benefits, the other one neither harm nor benefit. Okay? Remember I said that that was the easiest one because it's just a whole bunch of terms, right? You have to know the terms. All right, here we go. Oh, well, here's an example of, and something like this is going to be there, right? So if an egg salad sandwich sitting in a warm car for 3 hours develops 10,000 salmonella cells, how many more cells would result in just 1 more hour being left in the car? How would you set it up? Doc, you want to come up and set it up? I'll give you the formula. All you got to do is put the pieces in it. The final population is equal to the initial population times 2 as a constant of binary fission to the number of generations. Rock and roll, Doc. The salmonella generation is 20 minutes. Come back. So everything's right except the generation. Because it's 1 hour. How many 20-minute segments are 1 hour? Erase that 20 and put it in. 3. Yeah. Correct. That's the answer. CFU, do not, because I think it's cars or buckets or something. You gotta put the units. Okay. CFU after 3. After CFU, put CFU. Yeah. Good. That's the answer. Circle it, Doc. So if you leave it like that, it's perfectly fine. If you solve it, it has to be correct. Okay? Okay, when you pull down the... There'll be something like that on there. It's going to be there. Okay, here we go. Describe... Oh, this is the Doc. Doc, you don't get to answer this because you answered it last time. This is the Doc's favorite question. Describe how an amphipathic detergent or soap could be used as a chemical control instead of as it's usually used as a mechanical control agent. So tell me how an amphipathic soap works. Has a polar and a non-polar side. What is the non-polar attracted to? Everything on your body. What is the polar attracted to?
Water.
Right. So when you rinse with water, what happens? The polar portion pulls everything away from your body. And now you clean your body. Isn't that cool?
That's a big.
5 points for doing that.
5 points?
That's not bad. 5 points. Good luck. 2 sentences and 5 points? Okay, fine. Listen. Okay, I'm gonna tell you I don't think I did this. Or I might not, because I had it on there, but I think I took it out. But let's just think about this. Why would chlorine be considered more effective antimicrobial agent than ethyl alcohol? 1 sentence is all I need. What kind of time? Well, what do we call that less time? Contact time. So it needs less contact time, therefore it's more effective. Done. That's all I need. I don't need, well, if you look at the actual data, the data actually says that 50 parts per million will actually kill microbiome tuberculosis in 50 seconds. I don't need that. It needs less contact time, therefore it's more effective. Done. Because everything is related to the phenol coefficient. And even though I'm not going to be talking about the phenol coefficient, I'm talking about contact time. Okay? Now, use the above table to explain contact time using alcohol necessary for gram-positive and gram-negative. So now you're only looking at these 2 things right here. So you would say what? It's not rhetorical. I'm expecting an answer.
2 minutes and 10 minutes. So.
Oh yeah, but you can't just say 2 and 10. I'm like, what does that mean? You have to. It's what?
It's quicker on gram-negative.
Grandpa. Oh yeah, gram-negative. You're right. So it's more effective at gram-negative. It only takes 2 minutes, but gram-positive it takes a whopping 10 minutes. Therefore, alcohol is not that good. Right? don't go over to a hospital and say alcohol sucks. Because then they're gonna call me and tell me bad words, yeah. Yes, Kristen? Oh, you have to change your hand up.
Oh, no, I was
Oh, okay.
declining a call.
Okay, fine. Oh, all right, now, this is kind of what one of the matching looks like. It might be exactly what the matching looks like, I don't know, maybe. Or maybe there's something different, okay? Here we go. Thymine, which one does thymine fit in over here? It's not rhetorical, I'm expecting an answer. A DNA nucleotide, that's correct. Good? Okay. Ligase. Which one? 7? What- Not A. AD attaches DNA fragments to the ligand strand. That's what ligase does. Bacterial DNA. 1 chromosome, AC, correct. Semi-considered replication. E, that's correct. Parent stranded, used as a template. UAA, UAG, or UGA? Which one? AE, protein synthesis complete, that's correct. 5' to 3'. Which one? B? Yeah.
Yeah.
DNA replication direction, very good. Purines,
Contains 2
good. Helicase, A,
Mhm.
unwinds DNA. Thymine dimer,
Is that?
BC,
BC.
ultralight damage, or ultralight DNA mutation. Polymerase,
It's the last.
C, copy CNN. Okay, I wanna tell you this. When I give you matching like this, do not draw lines and expect me to follow the line. I want you to put the answer on the Scantron, right? There were several of you who didn't do that, right? Answers go on the Scantron for matching and multiple choice. Do not draw lines so I have to follow the line. I don't really want to do that. Okay? All right. Anybody have questions? Okay, here we go. All right, if I get, this is going to be on the test, like something like this. If I give you that, is that DNA or RNA?
DNA.
Why?
Because it has 2.
Okay, good. Can you give me the complementary messenger RNA strand? The answer is yes.
Yes.
Go. Stop right there. If it's AUG, you know it's right because I told you that I would only give you a DNA strand that's going to code for AUG first. Let's finish. Go.
G, G, C, A, U, U, C, C, G, U, A.
And UAA is what?
Stop.
So you know it's right. How many codons are there? 5. How many amino acids is it code for?
4.
Why is this gonna be hard? And then you're gonna have to use a codon table to tell me which amino acids they are. Okay? Describe the replications as relates to the following. Leading strand. Leading strand. Five' to 3' continuously. Lagging strand. Backwards in fragments, Okazaki fragments, and then it gets put back together. Good. Semi-conservative. 1 strand is conserved in each of the new DNA molecules. I like that. I like that a lot. Oh, looky here. You knew it was coming. Okay. What is 1?
DNA.
Topoisomerase. What's number 5? This is the light pink thing. Primates. Very good. 8. This is this fragment right here. Okazaki fragment. A, it's this, oh, my letters didn't come out. It's this guy right here. B, and B is this guy right here. Lagging. How do you know it's lagging? Because it's in fragments. Very good. Oh, Lord have mercy. So, I'm not sure if I'm gonna put this on or not. Haven't decided.
Put it on there.
Do it.
What's A pointing to? What's this thing right here? The backbone. Very good. What the E is pointing to a 5' or a 3'? 3'. Why? Hydroxyl. And the D is pointing to 5' or 3'? 5. Why? Phosphate. Very good. So how many hydrogen bonds are here? 3. Therefore, this is what base pair? G and C. And what is this right here? C is what? It's either guanine or cytosine. Which one? Cytosine. Why? Because that's a pyrimidine, right? It's got one nitrogenous ring. Thank you. This one, therefore, has to be the other guy. It's the purine, and which one is that? Guanine. Here's the other guys. 2 pairs of hydrogen bonds. That is a AT. What is that one?
Adenosine?
Or adenine. We don't really call it adenosine unless it's attached to a ribose and 3 phosphates, which makes it ATP. But if you said adenosine, I'd probably be okay with it. But it's adenine. And then this guy is what?
The U one?
Not the U one, because this is DNA.
Oh,
it's double-stranded. Thymine. Yeah. Uh-oh. Looky here. Don't do this one? Jeanette's saying, not that one. I don't know, but it might be there. But it might not. Okay. What is going on here? What is it called when when DNA gets copied by RNA? What is that called? Transcription. What is this thing that has been copied and now leaves the nucleus? A transcript or messenger RNA. It comes down here and it complexes with the ribosome. And what is this process known when you have a growing chain of these things? What is that known as? Translation. Very good. What's this firecracker looking thing? And what are these little things called? A growing polypeptide or an amino acid. Each one of those is amino acid. Okay? So that's a review. That's what I have for you. Okay? Any questions? 25 multiple choices, 25 matching 3 parts, and some short answers. But you've seen almost all the short answers, right? I don't know. We're gonna find out. All right. Anybody else have any questions? Let's go to lab then. Take 10 and see me in lab.